The United College Distinguished Visiting Scholar scheme is made possible by the generous support of the College Endowment Fund. Under this scheme, United College is able to invite world-renowned scholars to Hong Kong to give public lectures and meet staff and students of the college and university. And this year, United College has much privilege to have invited Professor Chen Yuan-tsong from Academia Sinica, Taiwan, as our distinguished visiting scholar in 2012 and 2013. Without further ado, I would like to turn the floor to the moderator of today's lecture, Professor Nelson Tan from Department of Chemical Pathology of the Chinese University of Hong Kong, to introduce Professor Chen and the topic of this afternoon. Professor Tan, please.
Good afternoon, ladies and gentlemen. It is my great honor and pleasure to introduce Professor Chen Yuan-tsong as the United College Distinguished Visiting Scholar of 2012 and 2013. Professor Chen graduated with a medical degree from National Taiwan University, and then he pursued further study and awarded a PhD degree in Columbia University, and at the same time he practiced medicine in both Taiwan and US. From there, he started an extraordinary career in medical genetics and has pioneered at least multiple breakthroughs during the last 3 recent years of advancement in diagnosis and treatment of genetic diseases. Early in his career, Professor Chen worked in many of the most prestigious institutes in the USA, like NIH, and later appointed a tenure position in Duke University. There, he headed the clinical genetics section and diagnostic lab since 1980s and became the chair professor in 1990s. And he also directed the Duke newborn screening laboratory, which set the standard for newborn screening, which we practice in most of the developed world nowadays. After 2000, Professor Chen returned to Taiwan and headed the Institute of Biomedical Sciences at Academia Sinica. After serving 10 years in that position, he recently stepped down from directorship, but he maintained his research team. So what I described in the last message does not fully reflect what achievement Professor Chen has made to the field of medical genetics. For a scientist, it is a lifetime award if he can make 1 or 2 breakthroughs or make 1 or 2 major discoveries. For Professor Chen, he made 4. So I don't have the word to fully describe what he has done, but yet I must try to do so. Now, we go back to the 1980s. At that time, we don't know what gene causes disease. So this is the era of finding the genes. So Professor Chen discovered the gene causing glycogen storage disease in the children. So these children come into a hospital with big tummy, but yet they do not have enough glucose to use. So they have problems. So Professor Chen not only finding out the gene, but also found a treatment. So nowadays we use cornstarch to feed these children. So we basically is based on Professor Chen's discoveries. And Professor Chen also break the curse of being having a genetic disease, one have no cure. So after using cornstarch, something that is available in the market, he invented a drug to cure another glycogen storage disease, which is Pompe, and he invented the enzyme replacement therapy. And the discovery of this enzyme replacement therapy was so extraordinary that now it became the content of a Hollywood movie, which featured Harrison Ford playing the role of the scientist. So tomorrow, Professor Chen will give you a full account of why Harrison Ford is chosen to play the scientist and what is the background of the whole movie. So after coming back to Taiwan, Professor Chen is work more on the translation of genetic into clinical use. So he recently discovered what genetic marker are the causes of adverse drug reaction. That means after given a drug, you feel even further sick. So this is the title of today's lecture by Professor Chen, Preventing Drug Toxicity with a Genetic Test. So ladies and gentlemen, please together with me welcome Professor Chen to the stage.
Thank you. Thank you very much for Neil's introduction. And certainly I am very honored and privileged to come here as a visiting scholar. And I'm going to deliver two lectures, and one is today's, probably a little bit more academic research, but tomorrow will be more fun. We'll talk about a movie. I came yesterday and also have a chance to look around the campus. And this is indeed a very, very beautiful campus and very lovely student. And this afternoon, actually, I saw this cheerleading competition. It's very, very creative, yes. Yeah. So it really makes me feel young again. Yeah. Okay, today, I want to talk about the drug toxicity. This is something we don't want it to happen. Okay, so by doing the research to find out the reason why some of us develop the drug toxicity, we try to prevent the drug toxicity with a simple blood test. Okay, so let's see, how do I do this? I see it. This one. Thank you. Yeah. So what is drug toxicity? I think is when you get sick, you go to see your doctor, and doctor will prescribe the medicine. Okay, and most often, you take the medicine and you feel better. But sometimes you take a medicine, but the medicine does not work, so you're still sick. But in the worst case scenario is you take the drugs, and your symptom, your disease, get much worse, and that is because you develop this side effect because of medicine. And that is we call drug toxicity. And in medical term, it's called adverse drug reaction. This abbreviate as ADR. So what is important to study of drug toxicity? First is this is actually the overall incidence of very, very serious that require you be admitted to the hospital. Actually, it accounts for 6.7% of all hospitalized patients. And second, some are very, very serious and become a fatal event. Okay, so this is actually the 4th to 6th leading cause of the death. So in addition to the patient's well-being, that this is also large impact for hospital cost. The 5 to 9% of all hospital costs are actually related to these adverse reactions. For the pharmaceutical company, they develop the drug, spend lots of money, billions of the dollar, and many, many years to develop a drug. And approved, and when you use in the human, many patients. Some of the patients develop this ADR, and the drug has to be withdrawn from the major market. So for pharmaceutical industry, this can be a really, really nightmare. So I mentioned that You go to the hospital and doctor prescribe the medication, but not all patients respond to the same drug at the same dose. So we all think it's a doctor give us a medicine, we take a medicine, we'll be okay. Actually, this is not the case. For example, there's a drug called statin. This is a drug when you have a very high cholesterol. Okay, so it's a very, very common drug that used in clinic. 30 patients do not respond to statin. And some of the hypertension, when you have high blood pressures, then you're using the beta blocker. 35% patients do not respond. For the old generation of antidepressant, only 50% respond. Okay. So in addition to not all patients respond to the same drug at the same dose, they also have inter-ethnic difference. Some drugs may work very well in one population, but not in others. And also, we know some patients develop the drug toxicity, and this is actually something we worry most. If the drug does not work, then we can change another drug, or we can bump up the dosage. But if something happens because you take a drug and you become very, very sick, this is something we worry about. So why there's a difference in terms of the drug response? I mean, what does it all mean? And Nelson mentioned I'm a geneticist. So this is the way I look for disease. Is virtually all the diseases, except maybe you're hit by the car. Virtually all diseases have a genetic component, and some disease has very large genetic component. The lecture I'm going to talk about, the Pompe disease, is one with a very, very large genetic component. So only a single gene defect that will cause the disease. And the most common diseases that we know about, hypertension, diabetes, the arthritis, and so on, those have a genetic component, but also has environmental component. Okay. Even for AIDS, we know AIDS is caused by the HIV virus. Yeah. So it's an environmental component. It's a large because you need to have HIV infection. But the problem is we also know that some patients actually have HIV infection, but clinically they are very, very mild. I mean, they don't get very sick. Okay. And we know it's because there are some of the genetics protect them from overwhelming of the AIDS, the disease. And what we're going to talk about today, that drug response, I think it's no exception. It's have some genetic component inside. So this. Okay, thank you. Okay. So I want to show you this slide. Actually, it's a slide made in 2001 that 2001 is a year that the first human genome, the drive sequence, it's not complete. 90% drive sequence being published. Okay, so that means 90% of our genome, we know the sequence and we know many genes. So at that time, the people already predict that pharmacogenetics will be the one that go to become the personalized medicine. So the idea was this: the patient, with all the patient taking drug. Most of the patients will be okay. You can treat it with conventional drug or the dosage, and they do well. Okay. However, there are patients, are poor responders, that with regular drug and dosage, they don't respond. Okay. So But this is okay. You can treat it with alternative. There are other drugs. You can treat it with alternative drugs, or you can bump up the dosage, and hopefully that the patient will respond. This is something we worry about. That the patient take the drugs, but develop the adverse drug reactions. Okay. So ideally, we want to know what is the genetic makeup, what the difference, why make you become a responder, a poor responder, or the one you will develop these ADRs. So this was back in 2001. It's about 10, 11 years ago. So there are many review articles published in Nature, published in Science, talking about this idea. Say, very soon, with your genes, then you can take one of these 3 times a day. Okay. And on that time, people also predict that one can preventing drug toxicity with a gene test. So when you go to see the doctor, you ask the doctor, doctor, first check my genomes, okay, then give me the right medicine. Then they also talk about the pharmacogenomic clinic. So there are many of these articles talking about this check my genome, then give me the best drugs. Okay, but that time, this was in 2003, 2 years after the genome, the drug sequence published. Still a lot about talking, but there are very few really data that show this can actually happen. Okay, so that time I think it's a helped more than hope. Okay, but I want to show you that today's talk is just there are more and more good examples that actually we able to using our genetic information to better to give the doctor can give you the best drug for you and the best dosage. I became interested in this the adverse drug reaction. It's actually is pretty new. It's only when I returned to Taiwan about 10 years ago. A lot of the doctors in Taiwan told me that this adverse drug reaction actually is very common and very serious in Taiwan. And this is some of the newspaper clip. Basically saying that you get a very common medicine for stomach illness, then that causes death, and some of the drug for common cold and cause the blindness. Yeah. So many of these examples in the newspaper, and indeed by epidemiology study, Taiwan actually has more of this ADR than other countries. So what is the reason why the Chinese people in Taiwan has so many of these adverse events? Well, I think there are 2 reasons. One is we say, well, it's because 我们中国人的体质 So maybe it's our body makeup. Okay, it's different. Okay, and another reason is because we take too many drugs. I think when we take not only the Western medicine, we also take herb medicine. Traditional Chinese is, we just simply like to take drugs. If you go to the hospital and the doctor say, well, go home, no, you're fine. I don't think you're happy. I think you want the doctor give you some medicine. So I list 2 reasons. I think it's our body makeup and also maybe it's the drug overuse. Okay. So I want to ask you about what is body makeup. And to me, as a geneticist, I see body makeup is our genetic makeup, is our genomes. Okay, so we try to see why Chinese people in Taiwan have more this serious adverse event, and the approach, I took is a genetic approach. Okay. But before I talk about the specific research, I want to tell you about this is what there are two types of this ADR. The type A is the one more common, and usually is a dose dependent. So the higher the dose, the more drugs you take, then the chance you will develop the ADR is higher. Okay, and we know that the drugs usually work, and they have therapeutic range, and you don't want to overdose. So if you overdose the drug, then you will have this type A ADR. Another type is called type B. This has nothing to do with the dose. Okay, we can just take a regular dose, but some people that will develop this drug toxicity, and this is what we call you are allergic to drug. You have a drug allergy, or you have a drug hypersensitivity. Okay. And it's called idiosyncratic because in the past we don't know what's going on, and was unpredictable. Fortunately, it's rare, and it was believed to be a polygenic model. And I think there's an example like penicillin and penicillin shock. Yeah. But today I'm going to focus on Stevens-Johnson syndrome, toxic epidermal necrolysis. Yeah. But there are other examples of this type B adverse drug reaction. Basically, this is you allergic to drug and you develop this very serious adverse event. And I put on question mark on this unpredictable and polygenic model because I want to show you at least some of this Stevens-Johnson syndrome, toxic epidermal necrolysis, and now also the DILI actually is predictable. Okay, and it's oligogenic models. So this is what I'm going to tell you about. I want to tell you about this genetic study of this very severe cutaneous mean skin, the ADR, this the particular Stevens-Johnson syndrome and toxic epidermal necrolysis. These two actually is the same disease but with different severity. The toxic epidermal necrolysis is by far the most serious this kind of adverse event. Still carry about 30 to 40% mortality rate. Okay. Stevens-Johnson syndrome is also very serious and about 5 to 10% mortality rate. I talk about the pathogenesis and I talk about the clinical significance. So what is Stevens-Johnson syndrome? I don't know anybody has any actually experience of this the drug allergies. I guess you are all too young that you probably don't take drugs. Yeah, but like my age, we always take some drugs. Cholesterol too high, blood pressure is too high, and usually when you have a drug allergy, is you have a skin rash and you have an itching. Okay. But these patients different. Yes, they do have a skin rash, but they are generalized all over the body, Okay, and also the hallmark of this disease is this. Eventually, they develop the blister. Okay, and this blister rupture is just like this. The skin kind of peeling off, like when you suffer from the severe burn, the burn injury. You have a blister, you have a blister, and this blister eventually rupture and skin peel off. Okay. So this is very, very serious, something we don't want to happen in our patient. So not only skin, but also the mucosa. This mucosa, this is the mouth. Actually, what we're not able to see is actually inside the whole the gastrointestinal tract, and also our airway mucosa also swollen and affected. So this patient actually suffer a lot. They're not able to eat. They're not able to drink, and they have a breathing problem. And this young lady took a medicine. This medicine called carbamazepine, also called Tegretol. This is a drug, still very, very common drug that used for epilepsy, the seizures, and also a lot of pain, neuralgia. Okay. And it's very good for one specific neuralgia called trigeminal neuralgia. But this young lady had a very severe, actually, the toothache. But this drug is very good for the pain. So the doctor prescribed the carbamazepine. And she developed, after several weeks, she developed this very, very severe adverse event. I just want to show you this. This is not just a skin disorder because they can affect the inside mucosa and has a breathing problem. And this youngster actually, because of all the airway swollen, okay, so eventually need a lung transplant. In Taiwan, there are many drugs can cause this Stevens-Johnson syndrome. Many drugs. There are over 100 drugs. But in Taiwan, the most common drugs that cause Stevens-Johnson syndrome is carbamazepine. Okay, this is the drug I just showed you. The young lady took because of toothache. And this drug alone, carbamazepine alone, actually already represents 26% of all patients with Stevens-Johnson syndrome caused by these single drugs. Then followed by phenytoin and some of the NSAID and allopurinol. This is the drug, another drug that's very commonly used when you have gout. Okay, the high uric acid and causing the gout. And also the incidence of carbamazepine-induced Stevens-Johnson syndrome actually very, very high. In Taiwan, is per million carbamazepine user, you will see 2,300 patients would develop this. So the incidence is 0.23%. And this is much higher than other population, especially compared to Japan, compared to Caucasian population. Okay. In Caucasian population, carbamazepine is actually very safe. You rarely see that carbamazepine causes Stevens-Johnson syndrome in Caucasian population. Okay. But it's different in Chinese, in Taiwan, and also Chinese in Hong Kong. This is the number one drug. Causes Stevens-Johnson syndrome. Okay. So our first study is to look why Chinese, Han Chinese in Taiwan, why they are so at risk for this carbamazepine drugs. So I don't want to, I really don't want to go over the detail, but this is a genetic study. So basically you just collect a whole bunch of the patients with carbamazepine-induced Stevens-Johnson syndrome. Then you need a control. Okay, the control are the patient took the carbamazepine, but no problems. So you compare the genetic makeup of this patient and also the controls. Okay. So at that time, we want to see what gene may be associated in patient. Highly rich associate with patient, but not much for the control. And nowadays, I think you are lucky. You just sequencing the whole genome, sequence all the patient's genome, and you sequencing the control, and you try to find the difference. But this was 10 years ago, so we still have to think about what possible candidate genes that may involved in these ADRs. So basically, we have two categories of this candidate gene: the drug metabolizing enzyme, because this carbamazepine and other drugs, they also metabolized by drugs. And they can be phase 1 enzyme, phase 2 receptor transporter, and so on. And also in the literature, actually, there are also already many suggestions. This allergy, drug allergy, is actually is an immune mediate reaction. So, of course, we look many of the candidate genes in this immune-related gene, including some of HA, T-cell receptor, and some of the mediators. But anyway, in short, I just want to show you the result. What we found is actually is a human leukocyte antigen. Okay, it's called HLA, human leukocyte antigen. But one particular variant, we call it allele. One particular allele, this is B1502 allele. This gene variant, actually, we only studied 44 patients at a time, and all 44 patients carry these alleles. Okay. How about the control? Those patients took carbamazepine, but no problem. It's only 3%. We only found 3% of this control carry the allele. Okay. And in our normal population is 8.6. So this gives you a very high, we call odds ratios. Odds ratio is the way you measure the risk. So if you carry this gene alleles, okay, B1502, and the doctor gives you carbamazepine, the chance you will develop the Stevens-Johnson syndrome is actually, this is called odds ratio. This is 2,500-fold. Then, if you don't have this allele. So, if you have this gene allele, the chance you will develop the Stevens-Johnson syndrome when doctor gives you the carbamazepine is very high. Okay. Then we looked this allele distribution, the frequency distribution among different ethnic population. Then you can see here, this is basically in Southeast Asian population. Han Chinese in Taiwan, and also actually in Hong Kong, Thailand, Singapore, Malaysia, and the Philippines, the Aboriginal, and India. But this is very much enriched in this Southeast Asia. If you go to the north, Japan has very low, Korea has very low, and in Caucasian population in Europe, in US, actually is almost non-present, and Hispanic and the African American. So this is allele is more in the Southeast Asian. And if you look for the China, it's interesting. It's more in the south part of the China. When you move north, this allele is lower. Okay. So this actually can explain why carbamazepine is very safe in European population, in Hispanic, and also in Japan, Korea, because simply they don't have these genes. Okay. So one drug is very safe in one population does not mean that drug is safe for all the populations. Okay. So basically, there are many studies that also confirmed our original finding. And basically, this is a genetic marker in Southeast Asia, and including the study from your university hospital, Dr. Mann and Dr. Kwang, Patrick Kwang, in Zhongda Yuan. Yeah, that basically this is indeed a Southeast Asian's allele. There are patients in the U.S. Also develop Stevens-Johnson syndrome with carbamazepine, but they are Asians or Asian descent. Okay, so this is genetics. If you carry the gene, if you move to the U.S., you will still develop the Stevens-Johnson syndrome. Okay. More recently, that in Japan and also in Europe, in European population, they found another marker is A3101 that is associated with their population. Okay, but the odds ratio is not as high as our Southeast Asian populations. Oh, okay. So I want to show you another drug. This is a drug called allopurinol. It's a drug widely used to prevent the gout in patients with hyperuricemias. Okay, and now it's also frequently cause of the Stevens-Johnson syndrome. And we did a similar study, and this again is interesting. That the gene we found for these different drugs, but causing the same kind of the disease, is also HLA-B, that locus, but it's different from the carbamazepine. It's 5A01 allele, and again, this R ratio is high. It's about 500. So the chance you will get this allopurinol-induced Stevens-Johnson syndrome is still very, very high. And these alleles, if you look for the distribution among different ethnic populations, now you can see it's not only in the Southeast Asian, but also you can see in Caucasian population, you can see in the Europe, and you can see in the African. Okay. Although the highest is still in the Han Chinese, and including the Hong Kong, mainland China, but it's not zero in other populations. So that time we also predict this allele is also will be important for other populations. And indeed, this is the case that not only in the Taiwan, China, Singapore, Japan also has a similar finding, and the European population also have a similar finding. Although it's not 100%, it's about 60% of European population is associated with this allele. Okay, so the next question we ask, so is this HLA is just a marker, or is really involved in the pathogenesis of the disease? Okay, and we actually now we have a lot of evidence that this is not a simple association. This is actually involved in the pathogenesis of the Stevens-Johnson syndrome. What happens is the keratinocyte, this is skin cells. The skin cell that carry this particular allele. Okay, here I'm using the carbamazepine as an example. And the skin cell with this HLA-B1502, then the peptide and the drug that bind to this HLA-B1502, then present to the T cell with specific T cell receptor, actually recognize the whole complex. And this is the immune-mediated reaction. So you activate the cytotoxic T cells. Then when you activate the cytotoxic T cell, you produce these cytotoxic molecules. Okay. I don't want to bother with you the experimental detail, but I thought I want to show you just a few slides and talk about this cytotoxic molecule, the granulysin. I think this is. I think you all heard about this, the fast ligand, porphyrin, the granzyme, and this is our cytotoxic molecule. But the way actually it has been shown, the prior study actually showed this, the fast ligand or porphyrin granzyme actually is a mediate the rapid disseminate the skin cell death in Stevens-Johnson syndrome. Okay. This was published in 2004 and published in 1998. But the important is in order for porphyrin and the granzyme B to work, is this cytotoxic T cell need to have a direct contact with the skin cells. Then they release this cytotoxic molecule, then kill the skin cells. Okay. However, when we looked at the actually pathology, and the pathology for this Stevens-Johnson syndrome, that there's a lot of the epidermal, the skin damage, but not many T cells, not many T cells. In contrast, those common drug rash, usually when you take the drug, you have a drug allergy, you have a rash. This kind of common drug rash. You have much more the T cells than the more severe this the Stevens-Johnson syndrome. So this does not make sense because there's a lot of the T cells than in the very mild skin rash. But very few this cytotoxic T cell in Stevens-Johnson syndrome. So we don't think it's a direct cell-to-cell contact. It's a very efficient mechanism, to cause this rapid the cell damage. So we did a study, and I just want to show you. This is we look for the blister cell. This is more of the cytotoxic T cells, and the very important one we found is a relative new cytotoxic molecule called granulysin. Also, we also found the granzyme B, but it's not as high as granulysin. Then, when you look for this, the blister fluid from the patient, that you can see the granulysin is very high, much higher, 1,000-fold higher than granzyme B, than porphyrin, than phospholigand. So we think this is the most important one. Yeah. And indeed, there are many ways we're able to prove this is indeed the most important cytotoxic molecules. I don't want to bother you with this experimental detail, but I thought it's important thing is now we have all this basic research. We found, we identify the gene variant, and we know the mechanism, but how about the patient? I mean, is this any kind of research to lead a better care of our patients? Okay, so this is so-called translational research. So we want to know is do this biomarker actually have potential to use for further development test? So we able to identify the individual at risk for this drug-related very serious condition. So in other words, can we preventing this severe ADR by screening people at risk before the doctor give you the medicines? And this is something we would like to see as a scientist, armed with a genetic tool, that we want to see whether we can do a better job than fortune tellers. However, I also want to show you there are many, many biomarkers in the literatures, and this is reviewed last year that in Nature. That basically saying, there are many, many, many papers. The estimate number of the paper document thousands of this biomarker, more than 150,000 of the paper, many, many thousands of the biomarker. But most of the biomarker actually have no clinical utilization. So this is a drop in the ocean. So we worry about, our marker, our gene is actually also drop in the oceans. But when we did the calculation, if we're using this B1502 as a test for carbamazepine-induced Stevens-Johnson syndrome, actually this test has a pretty high sensitivity, specificity, and if you are negative, if you don't have this allele, then you are very, very safe. You can take the drugs. Okay. And if you are positive, you have a very, very high chance. Although it's not all the positive patients will get the Stevens-Johnson syndrome. And now we know why. Actually, there's a T-cell receptor also involved in the process. So this is my theory. We think this will be useful clinical markers. Yeah. And our Department of Health actually agree, say this is important, that the doctor need to know this genetic association. And also the US FDA, actually they make a very strong recommendation. Basically the US FDA relabel the drugs, and with this information saying patients with ancestry from area in which HLA-B1502 is present. That is in Hong Kong, in Taiwan, in China, in Southeast Asia. And if you have a patient with ancestry from this area, then your patient need to be screened for the allele before starting the treatment with carbamazepine. Okay. So although FDA relabeled the drug, and this was 5 years ago, they relabeled the drug, but the problem is physician going to use it. Okay. That because physicians don't like genetic information. Physicians want to see is a real clinical data. So we end up we did a prospective study, actually to see if we can using this genotyping before the doctor prescribed the drugs, the gene test, whether we can really prevent this carbamazepine-induced Stevens-Johnson syndromes. And so this is designed. Basically, I say the study duration is 3 years because the government only give us funding for 3 years. And they asked us to do 5,000 patients in 3 years. And this is really a big challenge. In the lab, we can do any time we want, but in the real clinical setting, that this is what we need to show the government this indeed is have a good clinical use. And the reason for 5,000, this is our statistician come out the power. So just want to show you very quickly. So in the first visit, okay, we will draw the blood, okay, a little bit of blood after informed consent form. Then we will, the doctor will send the blood to us, okay. Then we do the gene test. And within 3 days, we report the result to the doctor. So if it's, the patient is negative for this gene variant, then it's okay. Then the patient can start the carbamazepine. But if it is positive, they will come back to the clinic. Then the doctor will discuss with the patient, and they may be using the other medication or something. Okay. So anyway, so we complete, finally we complete the study in 3 years. And okay, yeah, I'm running out of time. But just want to show you a result, and this is important to show you, is that although in these 5,000 patients, that there are some patients still have some adverse event, but these are the very mild skin rash. Okay, there are 7 subjects actually had more widespread rashes and were hospitalized. Okay, but none of them actually has a Stevens-Johnson syndrome. They have some the more widespread skin rash, but these are not severe as Stevens-Johnson syndrome. So we are happy. In all those 5,000 patients, we did not see any of Stevens-Johnson syndrome. So this is statistically is a very, very significant, because without the screening, then we would expect the 10 cases of the Stevens-Johnson syndrome. But with the screening, we see none of the patient developed the Stevens-Johnson syndrome. So this is in 3 years we collaborate with almost all the hospital in Taiwan, total of 23 hospitals, many doctors that complete this study in 3 years. And the lead PI is Dr. Chen. So anyway, this is the last slide. So I show you this HLA-B. It's very, very important. It's not only is a genetic risk gene, but also a major pathogenic road. These are the 2 I show you, but now they actually have many of these examples of the HLA associated with drug hypersensitivities. Yeah. And also, the risk for this drug hypersensitivity is likely ethnic specific. So some drug is very safe in one population, but may be very, very dangerous in others. And this is due to because whether this population has a risk gene or not. Okay. I mentioned about the granulizing. Then the last thing is this is the preventing drug toxicity with a gene test is actually now is a clinical reality. And I know in Taiwan and also in Hong Kong that now when doctor want to prescribe the carbamazepine, they will do the test to make sure the patient's safety. So this is something from the laboratory research that although the total we spend about 11 years, but eventually it's something that we happy to see there's some clinical utilizations. And this is lots of my students and also my postdoc and many hospitals. So this is the last slide. Just want to, it's a Chinese version of fortune teller. I want to wish you healthy, long life, but no disease. Okay. Thank you very much.
So thank you, Professor Chen, for the interesting and enlightening talk. So please be seated on stage, and I'm sure that there will be some questions from the floor. So may I now co-appoint Professor Tang, moderator of today's lecture, to come up to the stage and host a Q&A session for us. Professor Tang, please.
Thank you, Professor Chen, for the very informative lecture. And I know there must be many questions from students studying biomedical areas, but also for students in other disciplines, you are encouraged to ask questions because you just have a favor of what the genetic is coming in real life. So there will be a lot of ethical, legal, and even social issues. So you can imagine that one day you need to study all your genes, and put in a CD-ROM, and then carry the CD-ROM to see a doctor. Okay, this is my gene, and give me a drug. Okay, so I think that day will not be far away. So, yeah.
Actually, already happening. There's a company called Illumina. They actually have an Apple application, My Genome. You can download it. and you can put your genetic information in that, and you can show your doctor.
So, okay, so the discussion is now open to the floor, and any question is welcome.
Thanks for a great talk, Professor Chen. The question I asked is actually a pretty simple one. So you mentioned around 100 has been useful, or at least potentially useful for diagnosis, 100 biomarkers. So are they really in place now, or these are potential ones, or are they real ones?
The 100 marker, a lot of actually is cancer.
I see.
The oncology now using this target therapy. So there are markers associated with response to specific drugs. And also there are many markers associated with ADR. Yes, now it's useful. Yeah. And actually we just did, I just published a review article, and we list the clinical useful ADR test. Right now, at least there are 12 markers. Yes, clinical useful.
Thank you for a very inspiring talk. So if I remember correctly, the HLA test you mentioned, you said there's a positive predictive value of, say, 5%. That means that most people who have the HLA allele, even if they take carbamazepine, will still not have Stevens-Johnson syndrome. So what are the other factors which govern that?
Yes, this is a very, very important question. Is that even if you test positive for HLA-B1502, for this particularly positive, that they are still patient. You give the carbamazepine, but they are okay. And so this suggests there are other genes involved. So we did a GWAS, but we did not find other genes. But we found is specific T cell receptor to recognize the complex. So those patients, you need to have a HLA-B1502 positive, but you also need to have a right T cell receptor. Yes, and we just published that early this year. Yes. Yeah. Yeah.
So there is some sort of a T cell receptor rearrangement which can predict that. So if you plus that one plus your HLA test, then you might have a even more powerful possibility.
Yes, indeed. Yes, yeah. If you combine both, then it's much, much higher. Yes.
The reaction with allopurinol seems even more complicated because it's perhaps dose dependent or dependent on poor renal function. Is there some explanation for that?
Yeah. Well, it's actually something we don't know. Okay. The allopurinol that is much more complicated than carbamazepine, because in carbamazepine we only see CD8 positive cell. Okay. In allopurinol-induced Stevens-Johnson syndrome, some patients, a lot of the CD8, some patient has a lot of CD4, and a lot of patients has mixture of this, the CD, the two different kind of the cytotoxic T cells, and for the renal function, that certainly metabolites that accumulate. It seems higher. The oxypurinol actually is the one that we believe is the one causing this Stevens-Johnson syndrome instead of allopurinol. Yes. So So why the renal function is a factor? But when we actually analyze independent, consider with the renal function, the renal function is an independent risk factor. It's not something together with this allele. So we're not able to say, if you have 15A01 positive, but if you have the normal renal function, then you are more susceptible. No, we don't see that kind of correlation. Yeah.
Yes, Professor Chen, could you postulate why these particular alleles is only found in the southeastern part of Asia? And also the Chinese, there is multiple races. With the Chinese people, they are multi-races. With the different races among Chinese have different frequency of these particular alleles.
I was told that the human migration history, we all started from the Africa, then to the Middle East, and there are two. One go to the south, one go to the north. And part of the Indian, Thailand, Taiwan, is the one belong to the south. So somehow this gene allele has some advantage in certain stages, million years or something, that remained and go through this southern migration part. Yes. Yeah.
I see. Okay. Yes.
Professor Chen, I come from a different background. I come from engineering, and in general, my area, the larger area is information science. I'm curious as to whether what your thoughts may be on the current hot topic of big data. We talk about dealing with large databases, and in genetic research, whether you see any opportunities for convergence between information science and the kind of research you're doing.
Yeah. Well, we need a lot of help. I think it's now the machine can generate, all this billion of the, I don't know this. Anyway, the huge amount of the data, but the storage is a problem. Okay, so the people say, well, maybe we don't need to store the raw data, but you never know. I don't think we are ready yet to discard the raw data. So that storage, we need help. Okay. Annotation, analysis, that is not another challenge, and we just simply don't have people. Machine can generate, tons of the data. And right now, everybody is sitting on too much data, too little power to do the meaningful analysis. Yes.
So can you specifically give an example of how you do information filtering at the current state? When you talk about having too much data and you have to filter for some useful data for your research, can you give an example of how you're doing information filtering now?
The problem we have now, in order to do accurate sequencing, so for one genome, the 3 billion base pair, we not only sequence one genome 3 billion. We need to do 30 times coverage. So it's a 90 billion base, okay, for one genome. Then we don't know the accuracy. So we will need to have a way to determine on this, even though 30-fold coverage, statistically, if you see the difference, then are they false positive or are they real positive? So right now we have to do a lot of experiment there are some algorithm we can do, okay, but it's not good enough. So a lot of time is actually analyze the data to make sure this is real your genetics makeup. Yes.
Thank you very much.
Thank you. I have what might seem a stupid question, but I hope will be of interest to the audience. So if you say that this is a gene that's in a certain population of Chinese people, and if they intermarry with non-Chinese people, how many generations of their offspring will it take before this isn't a concern to that family?
I think it's just one generation you will see. This is dominant. You only need one allele. Yes, yes.
So it will stay forever?
Hmm?
It will stay a concern forever?
Oh, yes. Okay. Yeah.
Thank you.
Yeah.
It's the probability that it was Chinese.
50%, yeah.
So, yeah, we have a question in the back. Thank you.
Stevens-Johnson syndrome, is that mean that he has, all his family has a dominant genome alleles that he must take care before taking certain drugs like the allopurinol? Does that mean that he is necessarily has the risk to develop serious syndrome?
Yeah, this is a very important question. It's say if you go to a hospital, you test positive for 1502, then the doctor say yes, you are at risk for carbamazepine. So not only you, your sibling, your parents, one of your parents will carry this allele. Okay, so and your brother, sister have 50% of chance also carry the allele. Yeah. So that's what we recommend. You know, if you have a family member test positive, the others should be careful about this the same kind of drugs. Yes.
And is there any method to relieve or to lessen the syndrome of Stevens-Johnson?
Is there any?
It's the skin damage.
The skin damage?
Yeah. Can you relieve the syndrome?
Yes. Nowadays, once you have a patient with Stevens-Johnson syndrome, then it's you put in the intensive care unit. Or if your hospital have a burn unit, the burn injury, you put on the burn unit. Okay. So the first thing, the important thing is you don't want to have a secondary infection because the skin already peeling off. You don't want to have a bacterial infection. Then there are some of the, because it's an immune mediated, so there are some kind of immunosuppression kind of treatment, like give a large dose of steroid or some called IVIG, immunoglobulin. But even that still carry even nowadays still carry high mortality rate. Stevens-Johnson is about 5 to 10%. And 10 is actually 30%. Yes. Yeah.
Thank you.
So we don't want it to happen. It's better not to happen. Yes.
Hello, Professor Chen. Based your talk, this is about CTL mediated hyperreaction in this drug toxicity. Based what I know in why those patients develop drug toxicity, escape the clonal lesion in the T cell, and for example, in the penicillin allergy, most of patients they will have a clonal lesion to clonal deletion up on the cytotoxic T cell, so they pick up to eliminate the possibility to trigger the drug toxicity in human. So why those patients develop as the Stevens-Johnson syndrome, they escape the clonal deletion on the T cell, about the T cell.
Okay. Yeah, that's a very, very good question. I think this is kind of we can consider this is an autoimmune disease. Yeah, it's actually our body the T cell try to fight the foreign thing, and they look the drug as a foreign, like a virus, and they try to eliminate. Yeah.
Okay, so is there any more questions? If not, oh.
So, Professor Chen, I just want to ask on behalf of all the audiences here about your stand of the general screening of the genome sequence of us, of human, for clinical users and also for other users. So what is your opinion on that?
For sequencing
everybody. Sequencing everybody, provided that we have the bioinformatic tools and storage capacity to store everything, and every doctor can, every doctor or other people can use the data, and maybe that takes a lot of argument, but just for sequencing itself, do you think it would be nice to have that, or are you against that?
I really think it's going to the technology, and bioinformatic tool is getting better and better, and the really thing that may slow down is actually LC issues that may slow down our clinical utilization. Is how to protect the privacy. I think that is will be could be a biggest hurdle. Yes.
Yes, but my question was, what is your opinion? So, are you against it, or are you for it?
I'm for it. Yes, I think as a scientist, I think it's the more I know myself,
Well, won't one be scared to death if I know that I'm going to have a certain disease in a few days or a few years?
No, actually, I already have my gene sequenced.
Okay, good.
So I know that I have 10 risk factors for cancer and 2 risk factors for Alzheimer's disease. So I think LC issues means ethical, legal, and social issues. And there's we come back to movie. So a highlight for tomorrow's talk. So there's a movie a few years back featuring Tom Cruise as the actor. And he had, now, that is something like 2050. The world, everybody have sequenced your genome after birth. And then Tom Cruise do not have the gene to be something he want to be. Okay, so he have to get the fake identity in order to be something like a pilot. So will this happen? So these are all related to LC issue. And when whether the government want to know your genome before the government give you a certain job, and then this will be a real issue for coming years. And
actually, with Professor Dennis Lowe's advancement in research, actually we don't have to wait until we are born. So before we are born, we know our genes already.
Yeah. Okay. So any more questions from the students? You are most welcome to ask questions. If not, ladies and gentlemen, please join me to thank Professor Chen again for very.
So thank you, Professor Cheng and Professor Tan again. To express our college appreciation, we have prepared some souvenirs for both the speaker and moderator. And may I now call upon Professor Jimmy Yu, Head of United College, and Professor Helen Meng, Chairperson of the College Distinguished Visiting Scholar Committee, to present the souvenirs. Professor Yu and Professor Meng, please. Firstly, to our distinguished visiting scholar, Professor Chen Yuan-tsong. Then to Professor Nelson Tan, moderator of today's lecture.